Thank you. Good afternoon, everyone. Thank you very much for attending. It's a great pleasure to welcome Dr. Geraldine Walsh back to the CBR. Geraldine completed her PhD degree at the Royal College of Surgeons in Dublin, Ireland in platelet physiology, followed by a postdoctoral fellowship here at the CBR under the supervision of the late Dana Devine. Since her research training period, Geraldine has occupied several prominent roles at Canadian Blood Services, including the knowledge broker, and now is the lead development scientist at CBS. And we're very happy to have her speak to us today about plasticizer-free blood bag systems. Geraldine, thank you very much for speaking.
Thank you, Hugh, for the very nice introduction. And it's a real pleasure to be here and to be on this side of the podium, which I don't think I've actually ever been up here before, despite many years on the other side of the podium. So today, as you mentioned, I'm going to talk to you about a pretty significant transition that's happening in transfusion medicine, and it's coming to us in Canada over the next few years, and that's a move away from a plasticizer that we've been using in blood bags and other medical devices for many decades. I also wanted to take a moment at the start to introduce myself and where I work and the type of work that we do there, for those of you who don't know me. So I work at NECAD, which is just across Westbrook Mall from here, and it's a Canadian Blood Services development facility. And this is a small facility that includes a clinic where we can draw blood from predominantly deferred donors. We have a space where we can process that blood into blood components, and then we have testing facilities as well there, and also we do some of our testing here at CBR and in collaboration with folks here. So this, the focus of NECAD and the work that we do is very much applied development. We are looking for solutions, new products, new processes, optimizing our current products and processes in a way that allows them to then be operationalized by Canadian Blood Services. For example, if there's a new product that needs to be introduced to the fresh blood product portfolio, it'll probably start out in NECAD being looked at there. So it's very much applied development work that we focus on. So just to mention briefly a couple of disclosures, I am involved in projects that receive in-kind support. Example for this project being prototype blood bags from commercial companies, but I don't have any other conflicts of interest. And I thought I'd start with some learning objectives so everybody knows what to expect from my talk. So I'm going to hopefully give you guys an understanding at a high level of the history of blood bags and plasticizers in blood bags. By the end of this, you'll hopefully be aware of some upcoming changes that are going to impact blood bags here in Canada. You'll be able to describe the results of some of the development work we've done at NECAD, as well as be aware of some key considerations as this transition approaches. And my outline very much aligns with the learning objectives. I'll start with some background, then go into the development studies, and then the considerations. Okay, so a brief history of blood bags to start with. So a couple of years ago, the AABB published an abbreviated timeline of great moments in transfusion medicine history. And among them, they identified the moment in 1950 when doctors Walter and Murphy introduced plastic bags for blood collection. So this move replaced glass bottles. So up to that point, we were using glass bottles to collect blood and store blood. And the replacement with plastic obviously had a number of large advantages. However, it was more than just an advance in the material that the blood bag was stored in. This switch to PVC plastics actually laid the path for the beginning of what's known as the modern era of component therapy. So it allowed us to stop, to take blood, and instead of just transfusing whole blood back to a patient in need, to actually take that blood and separate it into its individual components, so red cells, platelets, and plasma. And this is because those plastic systems could be sterilized and could be kept flexible and could keep everything sterile. What made the PVC, the plastic, which in itself is actually kind of rigid and a bit brittle, suitable for this purpose was actually the plasticizer that was used with this. So a plasticizer is something that makes the plastic flexible, and it allows it to stay flexible in many different conditions and many different temperatures. And back in 1950, the plasticizer that allowed this breakthrough was a plasticizer called DEHP. And I'll go a little bit more into what DEHP is in a few moments. So as I mentioned, the change wasn't just a marginal improvement, it fundamentally changed how we do blood banking. And 70 years later, PVC DEHP is still extensively used in blood manufacturing and blood storage, although it has been phased out of many other areas. So it's truly everywhere within our blood banking infrastructure. And at Canadian Blood Services, it remains the plasticizer in our whole blood collection sets and in our red cell storage bags. And a survey a few years ago identified up to 47 consumables within our operations that involve or have DEHP. So what is this DEHP? It's a phthalate ester. As I mentioned, it makes rigid plastic flexible. But a key point with DEHP is that it isn't actually bound to the plastic. It's not chemically bonded to the plastic polymer. Instead, it sits within the matrix and it can leach into whatever that plastic is in contact with. So in this case, that's the blood that is in the bag. The leaching is influenced by several factors, including temperature, exposure time, and the lipid content, but DEHP is lipophilic, so it does readily move into fats, essentially. And it accumulates in the red blood cell membrane, particularly in blood products and also in plasma. And just to note that this isn't just a minor little coating on the plastic. It can actually, DEHP, the plasticizer, can account for up to like 50% of the weight of the plastic bag. So it's not an insignificant amount. However, there is a flip side to this, which is that phthalates are also known endocrine disruptors. In vitro studies and some animal studies, predominantly in rodents, the evidence in humans is not super strong, have indicated long-term potential adverse effects of exposure to DEHP, including reproductive toxicity and carcinogenicity. And this applies not just to DEHP, but also to its primary metabolite, MEHP. So as I mentioned, we don't have definitive toxicity data in humans. However, if you are a population or a person who gets a lot of transfusions, so somebody who's transfusion dependent, or for example, neonates, very small patients, these populations are going to be at greater risk of any toxicity that might result from this plasticizer. So with that in mind, why have we been kept using it in blood bags for the last 70 years? Well, what was noticed right from the get-go when Dr. Walter discovered this is that DEHP actually favorably impacts the red blood cell membrane. It stabilizes the lipid bilayer, and it reduces membrane fragility, and it reduces the hemolysis of red blood cells. So it's essentially the presence of DEHP in these plastic bags has really helped us to be able to store blood out to, say, 6 weeks. So red blood cells, for example, are stored for 42 days or 6 weeks. And this favorable impact meant that while DEHP has been removed, as I mentioned, from other areas of our plastic surrounding life, it still persists in blood bags and in red cell storage bags. And at this point, it's really integrated into virtually everything we know about red blood cells. So every stability study, every shelf life study, every clinical study, everything has taken place in the last 70 years in the presence of DEHP. So this balance, we've been living with it for a long time. And as I'm a bit behind myself there, it's really integrated into everything we know. So why are we talking about this now? Well, the European Union has introduced new regulatory changes that have tipped this balance that we've operated with for the last 70 years away from continuing to use DEHP and toward removing it and focusing on the potential safety concerns there. So essentially this legislation will come into effect in the European Union in July 2030, and it effectively bans DEHP in all medical devices, including blood bags from that date. And that applies to bags or devices sold within the European Union and manufactured within the European Union. So why are we talking about this in Canada? Well, Canadian Blood Services is part of this because medical device manufacturing is a global industry, and these effects will ripple worldwide. It's particularly relevant here for us in Canadian Blood Services because our component production processes follow those in Europe. So we procure our blood bags from European manufacturers. And we actually, our sole provider of blood bags right now is based in the European Union. So we will need to implement non-DHP blood containers as manufacturers can discontinue the PVC DHP products. At the moment, different vendors are exploring different plasticizer, non-DHP plasticizer options together with newer additive solutions, but it's only become clear in the last 3 years or so what might actually be coming to market as this change happens. So the good news is there is actually a lot of in vitro data in the literature now supporting alternatives to DHP. And many of these studies focus on red blood cell quality. As I mentioned, this is the cell that we think will be most affected by this change. And there were guidelines published a few years ago on how to conduct these studies. So what do we have in terms of options that are out there? So the main plasticizers that have been put forward to replace DHP are called DINCH. Hold on, do I have my pointer? Wait 1 second. There we go. DINCH, BTHC, DHT. There's some proprietary compounds floating around and also kind of combinations of these. And these are being put forward together with, as I mentioned, next generation additive solutions. But just to note from the outset that currently no DEHP-free whole blood collection set or next generation additive solution is licensed in Canada. But as I mentioned, there's a lot of in vitro data. I don't actually expect anybody to read this table, but I'm just putting it up there. It's from a review last year that kind of gathered together the expiry in vitro quality data on red cells. And the bottom line of this is that the main quality parameters, in vitro quality parameters we look at in red blood cells are looking fairly good in these various different alternatives with subtle differences between them. And I'm going to dive in a little bit more on a couple of the more recent and more relevant to the work I'm going to go into publications. So these actually 5 papers that I have here are quite relevant to what I'm going to talk about. And the overall conclusions of all of these are very positive in terms of the future of DEHP-free blood bags. So Gromysk et al. Found that while hemolysis was a little bit higher in DHT bags, if you used PAGZEM, which is a newer additive solution, it didn't look too bad. It looked closer to what the control already sees, which is today's. Larson et al. Found something similar. They said that DHT provides adequate blood component quality, and they also looked at irradiated red blood cells and found relatively good quality in irradiated red blood cells as well. This paper in 2024 looked at a DINCH PVC set, also found satisfactory quality in this DHP-free environment. And there's also this paper from Vermeulen in the Netherlands, and they found that the red cells stored in PAGZEM with BTHC, that other alternative I mentioned, also was quite good. This is an interesting system because it's actually a hybrid system. They have a different plasticizer in their whole blood collection bag and in their red cell storage bag. So one of the things that really is emerging in the in vitro data is the importance of pairing DHP-free plasticizers with newer additive solutions compared to the ones that we're currently using, at least here in Canada. So I just want to take a moment to take you through some of the hemolysis data from some of these papers to kind of illustrate that point. So hemolysis is a very important quality control measure of red blood cells. It's basically looking at how the red blood cells, how many of them have burst, which is obviously not a good outcome for red blood cells and their ability to function. And it is actually a controlled quality control parameter within our Canadian standards here in Canada. And it's also can be linked to product safety and product efficacy. So you want your hemolysis to be low. So you can see from, this is from the Gromysk et al. Paper, that they looked at DHP AS1 red cells. That's the current situation. AS1 is the name of the additive solution that's used a lot in the US. When they removed DHP but kept it in AS1, the hemolysis levels went quite high. But when they had the alternative plasticizer but added in this PAGZEM additive solution, that's a more advanced additive solution, the levels of hemolysis went back down closer to what we see today in our DHP containing products. And a very similar results. This is from Larson et al. So this study is a US study. This is a European-based study. And you can see it's very similar in the current additive solution and plasticizer. It's low. It goes up when you remove the plasticizer, but don't add in a better additive solution. But when you use the better additive solution, it goes down again to a level closer to what we see these days. And just to note, as I mentioned, hemolysis is regulated. This is the FDA's hemolysis upper limit, and in Canada and the European Union, it's a bit lower at 0.8% at the end of storage. So I'm going to dive in a little bit to the alternative plasticizers that I mentioned, and I'm going to focus now on DEHT, which is the candidate plasticizer that we focused on in the studies I'm going to go through. So DEHT is di-2-ethylhexyl terephthalate. So it's a non-phthalate plasticizer. It's structurally and functionally very similar to DEHP. However, it leaches into the blood components at a much, much slower rate than DEHP, and I'll show you some data on that in a moment. And it has a very improved toxicological profile compared to DEHP. And you can see these non-observed adverse effect limits are much, much higher in DEHT compared to DEHP, about 100 times. So this paper was published a couple of years ago. It involves authors from Mako Pharma, who's a company that makes red blood cell bags and whole blood packs. And they looked at leaching of DEHP, the old plasticizer, and its primary metabolite, as well as DEHT, the alternative, and its primary metabolite, into different components at different times during storage of blood cells. This is obviously a complicated graph, but the main point I want you to take from this is that the leaching is much lower. You can see, maybe if your eyesight's good, that the x-axes are quite different here on these two. So it's much lower in the case of MEHT compared to DEHP. So essentially, this reduced exposure is going to be good news for patients, because it's going to mean that they're going to be exposed to less of this, and this compound appears to have less toxicology issues as well. The trade-off is, though, that DEHT doesn't appear to have that membrane protective effect that DEHP has. So that is where PAGZEM, this next generation additive solution I've been referring to, comes in. So this, you can just see PAGZEM's constituents here. Beside it is SAGEM, that's the one we currently use in Canada. So it's considered a next generation additive solution. It's an isotonic solution, so it prevents the initial red blood cell swelling that happens when the red blood cells move from the anticoagulant, CBD, into the additive solution. And it seems there's good evidence that it better protects against membrane loss during storage, and it better protects against hemolysis. And this is, while it's new to us and considered next generation, it's actually been used in Germany for decades. And in Germany, they have a 7-week shelf life for their red blood cells, which is pretty unusual. Most places have 6 weeks or less. So this brings us to the actual work that I wanted to talk to you about, which is an evaluation of a DEHP-free blood bag set at Canadian Blood Services. So I already alluded to this, but we have a sole supplier of blood bag collection sets currently at Canadian Blood Services, and that supplier is called Mako Pharma, and they're based in Europe. So they have to develop a new blood bag. And they have been working on a prototype DEHP whole blood collection set, which is based on the alternative plasticizer and additive solution that I've been focusing on up to this point in my talk. And we've been working collaboratively with them to assess these sets in the context of Canadian Blood Services and our processes and operations. So I thought I'd actually start by showing a picture, because I've been talking about blood bag collection sets, but maybe some of you have never seen, if you've never donated blood, you've maybe never seen it in action. So these are the sets that we assessed. They're sitting on a tray for an overnight hold. You can see that this is the whole blood collection bag where the whole blood was collected into, and tucked behind it, secured with an elastic, or a whole bunch of other bags. So essentially, this is what I'm talking about when I talk about a collection set. It's the bag the blood is collected into, and it's attached by tubing to all these other bags in certain configurations. And that's how we process things into components in a sterile way through this closed system, so that at the end of the day, what gets into the patient has not been exposed to the air. We're not processing out in the open. And that's really why component therapy and plastics and plasticizers are so interlinked, because you couldn't do this in a glass jar. So I also wanted to set some background on what happens to whole blood collections at Canadian Blood Services, because this change involves our whole blood collection sets. So everything that comes from that is going to be impacted by this change. So When we collect into whole blood anticoagulant, we collect into an anticoagulant and then we cool it towards room temperature. It's usually an overnight hold. And then we process it through what's called a top and bottom processing approach. And this separates the whole blood into its components: red blood cells, which we're going to talk a lot about today, plasma, and buffy coats. The buffy coats are further processed and pooled together to produce a platelet product, which currently at Canadian Blood Services is then pathogen inactivated, but we're not going to talk much about this. The plasma can go for transfusion or fractionation, or it can be further processed into other products. The red blood cells, they have this additive solution added, and then they're basically ready to go, and they can be stored in the fridge for up to 6 weeks. The vast majority of whole blood collected Canadian Blood Services goes through this process. But occasionally, because we live in a very big country that has sometimes very bad weather, we have to activate a contingency process. And this allows us to put the whole blood into a fridge within 24 hours, and that extends our time to process out for another 24 hours. You can see here, the red blood cell has to be in the fridge within 24 hours. If we use this contingency process, we have an extra day to get the blood processed, but after it's been in the fridge, we can only get red cells and plasma for fractionation out of it. We cannot do platelet products out of it. So this is a rarely used, but very important when we need it, contingency process. The third fate for whole blood at Canadian Blood Services is liquor-reduced cold stored whole blood. This is a fairly niche product at this point, and I'm not going to go into that any further in this talk. So with this change away from DHP, although all may be somewhat affected, really it's the red cells we want to look into. Red cells are also further processed or transformed for specific clinical populations. So we make small volume red cells for small people. We do irradiation, we do red cell washing, cryopreservation, and combinations thereof. And these modifications are really important so that we can meet the needs of particular clinical groups, but they can be harsh on the in vitro quality of the product. So as we move away from having this plasticizer in the products, it'd be nice to know how these transformations are going to affect the red cell quality. So to get to the work that we did, we basically in an independent arm study, 2 independent arms, compared our current DHP SAGM sets to these prototype DHT-PAGM sets. You can see that the nominal collection volume of those sets are slightly different. This was a business decision because we were currently using 500 ml sets, but our target blood collection volume is 480, and we wanted to better align with our target blood collection volume to reduce the number of underweight units we throw away. Also, because more people globally use the 475 ml, so there's better contingency and resiliency in our supply chain process if we go that way. So you can see it's different plasticizer, different additive solution, slightly different volume, but our target blood collection volume remained unchanged. And as we set out to look at this, we had a lot of questions. Mostly, will we be able to even use these bag sets with our current processes and all the equipment we use in the blood bank to actually do component processing? Can we maintain the quality of the products? In particular, will our red cells still meet our current quality control criteria, which are set by the Canadian Standards Association? Will safety be maintained? What will this new additive solution do to the product characteristics? How will the membrane be impacted? And a big question for everybody with this transition is, can we maintain our 42-day shelf life? So to run into our main process results, I've kind of alluded to the study design already, so I want to just run very quickly through the results that we saw on this. The most important results, the kind of bottom line up front, is that we found that red cells in DEHP-PAGM and the new plasticizer are expected to meet all of our current Canadian Standards Association guidelines criteria. And they have criteria around levels of residual white blood cells, the hematocrit, the hemoglobin per unit, and hemolysis, the quality measure I've already discussed. So both systems showed good 100% compliance across all of these criteria. So we should be able to continue to meet our quality criteria, and we did a tolerance interval bound analysis, and we were able to meet that with 95% confidence. I'm going to run through a couple of slides where I'm comparing a few different parameters with DEHP-SAGM and DEHP-PAGSM shown beside each other. So as you can see, not surprisingly, given that the nominal volume of the bags was different, we end up with a slightly lower red cell volume. But importantly, there was no difference in the hemoglobin dose. So it was 52 grams across both conditions, which means that we can maintain the same dose of hemoglobin going into the recipient across these bags. We did see impacts on hematocrit and mean cell volume, but these are predominantly related to the isotonic nature of the PAGSM versus our SAGM additive solution, and we don't expect these differences to impact the quality of the cells. And this has been shown in the literature before, that the different additive solution has different impacts. We saw some interesting things metabolically. So we did see ATP levels ending up slightly higher at expiry. I should have mentioned this is all expiry data on day 43, one day after the products have expired, so one day after 6 weeks. And this suggests pretty good metabolic preservation in these units, slightly better potentially than in our current units, and that might be related as well to the newer additive solution. We saw indications of slightly faster glucose consumption in the DEHP PAGSM packs, which is interesting because it's actually higher levels of glucose in the PAGSM additive solution to start with, but they still ended up with lower levels of glucose. But this didn't appear to translate into any sort of increased lactate or changes in pH. Importantly, we found that there was no statistically significant difference in hemolysis or levels of supernatant potassium ions between the two conditions. So hemolysis, I've already alluded to how important this is as a quality measure. Supernatant potassium is also very important, again, especially clinically. And this just shows that our findings are in line with some of the other studies that have been published on this, that have shown either very small increases in hemolysis or no increase in hemolysis in these new plasticizers when a newer additive solution is used. Okay. So interestingly, we did see some differences in the ability of the red blood cells to deform and their rigidity. So these were measured using a Lorca device, which actually measures the response of the red cells in different and the osmotic fragility of the red cells, although I'm not showing all the osmotic fragility data here. So in some ways, this was novel in our study in that it's the first time it's been empirically shown using Lorca, the difference in deformability. So the results show that in DHT PAGSM, the red cells are less deformable and they require, the KEI is higher, which means they require more force to deform. They're more rigid. However, these data are fully in line with everything that's been known about DEHP and how it affects red cells for decades. What we can't answer from these data, we can't relate these numbers to what might happen to these red cells when we transfuse them into a recipient and how they're going to, say, get through the microcirculation. That's something that unfortunately there's not much data to support that translation from these data into what's going to happen clinically. So to summarize this study, which was focused on our main process, leukoreduced red cells, we found that this prototype set was fully compatible with our main top and bottom component processing method. We saw no failures against our standards, and our statistical analysis indicated that these products would likely continue to meet standards if we were to move to DEHP PAGSM. The in vitro data support DEHP PAGSM as a feasible alternative to DEHP SAGM, and very importantly, that a 42-day shelf life remains appropriate for these products, so we're not going to have a loss of shelf life. However, from the deformability and some of the other parameters, there's signals that the red cell in vitro quality is impacted by the absence of DEHP, and that the use of PAGSM as a next generation additive solution doesn't really fully compensate for that loss of DEHP's membrane protective effects. So we did publish this last year in Voxang, if anybody's interested in learning more. So we then went and repeated the exact same study design, but using red cells produced by our contingency process. So although this is rarely used, nonetheless, we had to evaluate it to make sure that we could still produce good enough quality products if we were to use the contingency process in the absence of DEHP. And overall, we saw very similar trends in the data. We saw no significant difference in the hemoglobin dose. We saw the differences in hematocrit and ATP that we expected from the change of additive solution. We saw no significant difference in hemolysis and supernatant potassium. However, we did do that same tolerance band analysis to determine how well we could continue to meet the current Canadian Standards Association guidelines. And we couldn't actually get to 95% confidence that we would be able to meet the specifications of this requirement. We were able to get 80% confidence, but not quite. No, we could get 95% confidence that 80% of the population would, but the specification requires 90% of the population tested to meet the specification. So again, these findings highlighted the need for more work to understand the impact of removing DHP on cells that undergo harsher processing conditions, because ultimately this contingency process is harder on the cells. They're held for up to 48 hours and they're held in the fridge, so they definitely have a slightly rougher road than the main process. So I now want to move on to the next study that we tackled on this front, which was to look at irradiated red cells. So as I mentioned, this is one of the key transformations that we do to red cells to be able to meet clinical needs. So this was our study design. Again, it was 2 independent arms. This is more of a pilot study. The N numbers are quite low. We irradiated red cells that were collected and stored in DEHP-SAGM and in DHT-PAGSM on day 14 post-collection, and then held them out to day 28, so day 14 post-irradiation. There's time guidelines around all of this, and those added together are the worst case. That's like irradiating them at the latest day you're allowed to irradiate them and holding them to the latest time you can hold them, which is, we often do that in our development studies, and especially in these kind of pilot studies, because we want to see what's the worst we're going to see in terms of quality in these products. The irradiation of the red cells? Not really, no. What is it? So it's to basically to mitigate trolley and other kind of adverse transfusion reactions that might occur in certain populations. Yeah. Now that said, these are all leukoreduced products, so there should be very few white cells remaining in them. Yeah. I think they do it a lot for very vulnerable populations as well, like neonates or populations that they're particularly concerned about. What clinicians do we have in the audience? Cedric, of course. Yeah. Okay. So again, with this study, as I mentioned, we used this kind of worst case scenario, and then we did in vitro quality testing 1 day post that expiry, just to see how the red cells were looking. So I'm not going to go through all of the data that we gathered on this arm, but just to give you some of the highlights, we did see a statistically significant difference in hemolysis in the absence of DEHP. However, it's still sitting at a level that's below the guideline or specification, which is actually set not for irradiated red cells specifically. This is just the general guidance for red cells. We also saw an increase, a trend towards an increase in supernatant potassium levels, but they're still below what you might see in a day 42, like at expiry red cell that hasn't been irradiated. So there's no kind of safety concerns there. We did the same deep dive on our main red cell study, but I haven't shown that, but I wanted to take you through it with the irradiated red cells on the deformability and rheology differences in the red cells in the absence of DHP. So you can see that deformability was also compromised in the irradiated red cells when DHP isn't present, and rigidity is also greater. So essentially they are less deformable and need more force to deform. So it's the same as what we saw in the main study, but it's a bit more emphasized, I would say, in the irradiated cells. We also took them through Osmoscan on the LORCA, which basically measures how the red cells respond to osmotic stress. So there's 4 different parameters that this machine provides. So O min is the level at which they, and you can see it here in these cartoons on the side. O min is the osmotic, the osmolality at which they start deforming optimally. And you can see that this is lower in the DHT-PAGSM red cells. O max is the osmolality at which they stop deforming optimally. It's also lower in the DHT red cells. The O hyper is the half maximum elongation, also lower. And the delta O is the difference between the O hyper and the O min. And that's also lower in the DHT-PAGSM cells. So I'm going to take you through how we interpret these results. So essentially, since O min, O max, and O hyper are all lower, this tells us that this curve has shifted to the left for the irradiated DHT-PAGSM red cells. So there is a difference in the ability of these cells to respond to osmotic stress. And since the delta O is also lower, we can conclude that the range over which they can optimally deform, the range of solution osmolalities over which they can optimally deform, is also a little bit narrower, which suggests they have less range over which to basically adapt to osmotic stress. And again, with these data, there's not much known about how to translate these into what might happen clinically, or if there is a strong correlation to these results to what might happen. But in terms of in vitro data, there's a clear signal there. So what does this mean? Overall, we found that the in vitro quality of the irradiated red cells remained acceptable. Values are still below those observed in non-irradiated red cells at expiry. We did see the impact of the absence of DHP on hemolysis and deformability, as we saw in our main study. There was, as I mentioned earlier, one other paper published on this a couple of years ago, and our conclusions align with the conclusions of Larson et al., which found that it's suitable, but you're definitely seeing a little bit more impact of the fact that this plasticizer is no longer present. So again, the findings just indicate to us that we really need to look into this and keep an eye on this as we move towards this change, and to assess the impact of storing irradiated red cells in DEHP-free formulations, especially if there may be a little bit, if the red cells are a little bit less happy or more stressed or undergoing other changes. So we actually have a manuscript drafted on this that should be submitted next week. So what's next for us in this area? Well, I just flew through the irradiated data with you. That was the transformed red cell product that we started with because it's the simplest. It does not require any extra consumables. The red blood cells stay in the bag that you store them in, and then they're irradiated in that bag, and then they go to the patient. And there's no change in additive solution or anything like that. The other transformed red cell components that I mentioned earlier are a little bit more complicated, and we have embarked on pilot studies on these, but we're facing a lot of unknowns. So washed red blood cells, which are also used for specific conditions, for an example, patients who are IgA deficient and who might need IgA to be washed away, and cryopreserved, that process is used predominantly for patients who require, for storing cells for patients who require particularly rare phenotypes that might be hard to find in the moment. Both very important transformations to meet clinical needs. They both use something called a cell washer, the ACP215 red cell washer, and it comes with sterile disposable kits. And these kits essentially plug into the machine and you go through the process and the cells flow through them. All of these kits contain DEHP, and the manufacturer is not European based, and they have not seen fit to commit to us whether or not they, or commit to anybody, whether or not they will change or not change from DEHP. So we have done preliminary studies looking at these in what we imagine the state would be, where we might be collecting into DEHP-free bags, but then washing in DEHP-containing bags and storing in DEHP-containing bags. So as we go into some of these other studies, these are the kind of considerations we have to think about. The impact of transforming red cells and storing them as they move between these different plasticizers and potentially different additive solutions? Will it be acceptable? And more so, I think the types of questions we're facing is, how long can we store these for? Are we going to have to change all of our guidelines on how long the product is good for because of these kind of changes? When there's additional consumables needed, like in the case of the ACP215, will the vendor move to DEHP-free options, or will we have hybrid manufacturing processes within Canadian Blood Services? And will we have to find ways to modify either, as I said, timelines or other things about the cells to maintain quality? The other thing is the additive solutions need to be available in a certain format in order to be used with these systems. And we're not sure, currently there's no, say, PAGSM is not available in that format. And then, as I mentioned, the cryopreservation is about storing very rare red cells. So there's stuff in the freezers 10 years old. So we're going to always be in that case with the cryopreserve for at least the foreseeable future, where we've got things in DEHP and we have to figure out how they're going to look if, say, we do move to a DEHP-free, and then they're washed and stored in DEHP-free. And the thing about some of those rare ones is that they may be from patients with membranopathies or other interesting things with their red cell membrane already. And this is, of course, all happening around the red cell membrane. So we don't know exactly how that's going to go. So I just wanted to end. I'm going to go quite quickly through this because I'm realizing I am talking a lot, just like I said I would. So you might be wondering what clinical data exists. I'm working in the lab looking at the in vitro stuff. So what clinical data exists for these DHP products? Well, right now, there's not a lot out there. There has been 2 hemovigilance studies from the Netherlands, from Sanguine. The first one was kind of an early hemovigilance study collected as a prospective observational cohort study using, you can see the numbers here, 1,650 non-DHP products and over 5,500 DHP units transfused. And they found no difference in transfusion reaction rate between the 2 groups, and they concluded no signal of increased reaction rate. So this is just looking for like safety hemovigilance. Is everything okay? They did a follow-up study, which was published late last year. And they found, this is a quasi-randomized observational multi-center study, using the same non-DHP bags as before, but much higher numbers, well, relatively higher numbers, 7,500 DHP-free compared to over 25,000 DHP containing. And they did find a signal of increased transfusion reaction rate in the DHP-free arm, but just to note, they were all non-severe, grade 1. And they also noted in their study that that it's not clear if they were actually powered to really detect these, because they are actually, the actual raw numbers of transfusion reactions are really, really low in both arms, which is obviously a very good thing, but means that they may not have had enough power to really understand what's happening there. So this tells us, and I'll come back to this in a moment, that hemovigilance and monitoring large numbers of transfusions are going to become very important over the next few years as we move through this change. Just 3 weeks ago, the very first in vivo 24-hour post-transfusion recovery data was published on DHP-free components. These were processed and produced using a novel system that we're not using yet here in Canada. It's called a Revio system, using non-DHP bags. And they have a proprietary blend of things as their plasticizer, so I can't really speak to exactly what it is. But the good news is that they did 24-hour post-transfusion recovery in 24 healthy donors who were transfused at day 42, so at the expiry date, using Biden-labeled autologous red cells, and everything looked very good. They were able to meet both the European, that's the EDQM criteria for 24-hour post-transfusion recovery, and the FDA criteria for post-transfusion recovery. So their conclusion was, and there were no serious events or unexpected side effects. So they concluded that it's a viable and safe alternative to DHP containing sets. So as I mentioned, that study, which came out, I think, on the 10th of February, is the first in vivo recovery study to date on DHP-free red cells. And the data look good, so that's good news. There were lots of guidelines published about 5 years ago on how to do the in vitro testing to understand this transition. And they're very well established. But as I've kind of alluded to, depending on what you're looking at, and this is this is a general challenge of the field, it's very hard to translate in vitro quality data to what's going to happen when you transfuse these. So we need to maybe have, especially as we're seeing some signals of differences, maybe look into some specific indicators of the red blood cell function, like how well it's able to carry oxygen and move through the vasculature to deliver that oxygen. Late last year, the BEST Collaborative also published recommendations on this transition, and they concluded with favorable in vitro data gathered using these established guidelines, pre-market clinical studies may not be warranted, provided established and licensed plasticizers and solutions are used. And they conclude that a lot of the plasticizers and the solutions we're looking at are well studied. There's a lot of information available. So they're suggesting that maybe pre-market studies may not be needed. However, the importance of hemovigilance and post-market surveillance is very clear in this. And so I just wanted to take this moment to mention, and some of you might be aware of this, that in a case of horrendous timing, honestly, hemovigilance in Canada is currently under threat. The Public Health Agency of Canada that funds and houses our TTISS system discontinued funding, and so they're going to sunset it on the 31st of March 2026 without any consultation. So just as we're about to embark on possibly the biggest but probably quite subtle change in how we manufacture and transfuse red blood cells, we may not have a solid hemovigilance system. That said, there has been a lot of advocacy work. Andrew Shee, I think, has been leading this. And as last month I saw there was a letter published, they are going to bridge till the end of March 2027. And I think there's now, from what I understand, consultation happening that might hopefully have us come out with a solid hemovigilance system. Because personally, I think we need hemovigilance always, but it maybe has never been more important than right now as the world embarks on this. We've had 24 people transfused with autologous DEHP red cells so far. So I'm going to end with some considerations on how we're all going to navigate, especially those of us in the blood service, this increasingly complex landscape. So a reminder, there's currently no regulatory requirement here in Canada for us to remove DHP from our supply chains. This is all coming from Europe. Now, granted, California and Pennsylvania have introduced state-level legislation, so I think we can expect that this is the way things are going. But right now, there's no requirement for us to remove DHP from our supply chains. But we're facing then this situation, unlike our European colleagues, where we actually have choice potentially, and a lack of clarity from our non-EU vendors on whether they're going to move away from DHP or not. So it's likely that we're going to have this interesting interim state in Canada and in other non-European blood operators, where we're going to be moving things around in between different plasticizers. And it'll go from a situation where for the last 7 years, pretty much every red cell you could pull off the shelf was in DHP. Now it might be in DEHT, maybe it will be in DEHP, maybe it will be in something else. We're also, and appropriately so, facing resiliency and supply chain challenges and thinking about these. So as I mentioned from the outset, we currently have a sole supplier of blood bags, and that's not a great position to be in. So we're also at the same time, the organization is pursuing a dual vendor strategy, which I think is a good idea. But as different vendors are now developing different plasticizer additive solution combinations, it just could get more interesting than it was before and more complex. But as I said, we're not alone in this. So to end with some practical implications, this is still a few years away, sunset in 2030 in the European Union. I may have come over a little bit negative in all of this, but I don't mean to. All of the in vitro data, all of the data we're seeing so far is showing that this change will be safe and this change will be okay, but there's probably going to be subtleties that we would like to be aware of. And the vendors are working very hard to provide viable alternatives to DEHP in response to this change in regulation. The early clinical data is also very solid or looking promising, but it's just, that's not a lot of data so far. And I want you to be aware that in the future, in Canada anyway, we might have a DEHP-free-ish kind of situation rather than a fully DEHP-free. And it's going to be a bit more complex. And I know a lot of you are potentially doing research on blood, so I kind of want you to be aware of several things. One is it might come to a point where, depending on what it is you're looking at, I know some people here do look at deformability, for example, things might shift slightly if you're using blood cell or blood products from Canadian Blood Services. But there's also a huge opportunity here for basic research to understand this shift and maybe look into what exactly is happening at the red cell membrane, or, you know, with all these types of changes, there's always opportunity. So I think it's an interesting time, but it'd be nice to have a little bit more data. And with that, I just want to acknowledge this is obviously a huge team effort by Canadian Blood Services. These are the folks on the non-DHP project team. So Anita, who's based in Edmonton, the Associate Director of Development. We have Tatiana, Carly, Manu, who's in the audience here, Sanaz, Ken, we have Canadian Blood Services supply chain colleagues, including Wanda, Shelley, and Heidi. We have Zhang Ming here too. Your name's not on the slide. Apologies, Zhang Ming, but you've also been doing testing on this work too. And we did work very closely with some colleagues from Mako Pharma, Chrysalan, Stéphane, Martin, and Mathieu. And of course, nothing happens without the donors that come into NetCat. So I'd like to acknowledge them. And I'm open for any questions.
Thank you very much. Are there any questions? Chris?
Super, really, really nice and pretty interesting. So is DHP, the P1?
The P1, yes.
The removal of this by the EU, that was presumably not because of this product, but because of general long-term exposure from a whole variety of products and things, yes?
Well, so the thing is that it has actually been removed from a lot of different plastics over the last couple of decades, but they always exempted medical devices. And they were always exempting medical devices specifically with the effect on red blood cells in mind. So it's basically just now this regulation change is just aligning to everything else. There's not much DHP floating around in many other plastic products anymore.
But there's studies in for estrogen sensitivity and so on, was presumably based on chronic exposure rather than single-use exposure. Or I know we've got long-term transfusion patients and so on.
Yeah, absolutely. Actually, to be honest, all the toxicology data is in rodents. So it's a theoretical risk in many ways.
So probably along the same lines as Chris, but 40 years ago, when still the Red Cross with blood products, I was commissioned to try and study whether the DEHP was of concern. And my problem was I couldn't actually find any hard clinical data at that time. There was theoretical data, which actually presented in one of your early slides. So have you found a solution to a non-problem?
Not me. No, I hear you though. There is still no definitive clinical data in humans. So it's good to hear that you looked into that 40 years ago, but I'm sorry to say that situation hasn't changed. It really is about this theoretical risk, and it's the EU, it's really this legislation that's pushing this change, because the EU has made this this regulation, and there was a lot of pushback, as you can imagine, on this. And actually it was supposed to go into effect, I think, May 2026. And the industry pushed back on that a lot too. So now it's not going into effect until 2030. But yes, it is kind of switching a theoretical risk for a potentially safer product.
So one issue is the tail wagging the dog. And the EU is absolutely militant about any form of additive to any food stuff.
Yeah.
And that's been going around for years. They've obviously titrated this into the blood system. I have to see where it goes. But anyway, great talk. Thank you.
Yeah, and I mean, it's a really good point. And I think one of the challenges we face in this industry as well is a lack of options. There's not that many blood bag manufacturers. So it's not like you can just go out and be like, okay, the Europeans are out, let's go talk to somebody else. There's not that many companies in this area. It's not that profitable, I suspect. No. There's American, I'm sure, or people that certainly have plants in the States. A lot of the plants are based in either Asia or in Europe. Yeah. Hong.
So when you look at the in vitro data, there's definitely a lot of differences between the DHEP and the PEGs. Now, is there a way for anything to pop out that would be sort of disqualifying? Like you would look at the in vitro data and say like, okay, we can't use this.
Not in our data, luckily. I think there would, like if the hemolysis levels were very high or were exceeding the regulated levels, then absolutely that would be disqualifying. But we didn't see any of that, nor has anybody seen in any of the other studies that have been done to date. So that's what I mean by the in vitro data is looking very promising in many regards. However, hemolysis does seem to be a little bit higher. So it's really, I don't think there's going to be any issue with the vast majority of red cells, but it's when we add in some of these other things like irradiation or other things that it might start having cumulative effects that maybe we'll get to a point of meaning, not that we can't use these products anymore, but maybe we can irradiate up to day 14 and then store them for 14 days will become we can irradiate up to day 10 and store them for 10 days. So it's these kind of changes that I think are really going to come as opposed to any dramatic changes. But I do think it's what I would love is if we had a way to translate, say, the deformability numbers and data that we see into what will happen when you then transfuse those red cells into a person. But
we have some of that data.
I was about to say,
yeah.
I know that the work that you're doing might get us closer to that than what we have, with at our disposal at Canadian Blood Services. So there's plenty of scope for some good basic research in this field. Yeah, we should talk.
Yes, thanks.
Ed.
Hi, Geraldine. Deformability differed between the DEHT and DEHP produced red cells. Now, I'm wondering if that has to do exclusively with the compounds that are leached into the bags. DEHT has a slower leaching rate than DEHP. So I'm just wondering if we can be measuring those compounds in the membranes. Do we know that? We need a good person who's skilled in metabolomics to do that mass spec analysis for us, I think.
I may have a whole bunch of samples in the freezer upstairs, and I would love to get the opportunity to analyze them. So I do actually have samples and would love to do some like metabolomics and lipidomics to actually see what's happening at that level. I haven't got around to it yet, but it's a great question. And I do think this changes. I think this is very much focused on the membrane. So I think that a lot of the changes we're seeing with, say, the PAGSM, that's like metabolic changes. That's the additive solution and red cell metabolism. But ultimately, DHP leaches from the bags and intercalates into the lipid bilayer of the red cells. And DHT leaches less, and I don't actually know what it does or if it does anything. So I'm 100% sure there's going to be differences at the membrane that will be discernible.
Given that DEHT is a slower leacher, you would argue that, you could argue that those membranes are more normal if they're uptaking less.
I love this point because I like to say that we've been transfusing plasticized red blood cells for 70 years, and we're about to start transfusing not plasticized red blood cells.
And the real control there would be stored red cells in the absence of plastic.
Yeah, we basically have been using this plasticizer to influence the system. It's not natural. It's not our native red cells. So then, like, when you think about deformability changing, it could be going back to the deformability of the red cells that are flowing about inside.
Would that be good?
I can't imagine it would be bad, right? I don't know. I can't imagine it would be bad. I do think it has more implications potentially for storage and manufacturing and these kind of things. But I don't know. But then, I mean, we see that the largest hemovigilance study, we did see some indication of increased, granted mild, transfusion reaction rates. It's just, I'd love to have more data, especially in the clinical realm.
So I must have thought about adding DHEP as an additive in very, very low amounts, not just to rely upon leaching, but just as an additive to go into the membrane, then the bags can be DEHP-free, but there's already the free stuff there in the membrane.
I don't know that anybody's thought of that before. I've not come across it. So, although I suspect you'll have a hard hurdle to say, hey, can we add this potentially toxic thing we're removing back in? But no, it's yeah, in principle. And it
was a significant clinical effect.
Yeah. Yeah. Yeah.
That could be one way out.
Potentially. Yeah. We'll see. People are always trying to develop new additive solutions. So, and I think again that there's huge scope for improvement in the additive solutions too.
As in high frozen. Very nice.